So excited to see so many people here on the live. I've opened up our live lesson today for everyone who wants to do a little revision session before tomorrow's prelim. I know a lot of you are also probably still writing your prelims maybe in other provinces, but I know tomorrow is the Western Capes. This lesson I've opened up to every subscriber, not just the members. So that you can see what we do in live lessons, and maybe if you're interested in joining the membership, you can see what we do and how we cover different things. I'm going to jump right into it so that we get the most out of this lesson right now, and I'm going to be revising all sorts of things for paper 1. I'm actually going to start with plant hormones because I'm pretty sure it's the topic you are ignoring the most, probably, before the exam tomorrow. I'm going to go over some stuff. If you'd like to ask questions, you can put it in the chat. Just remember, everybody, that it takes me a little bit of like a hot second or two to read the chat. I might even actually, now that I think about it, I will open the chat alongside me on my laptop so I can actually see what everyone's saying simultaneously at the same time. Let's just have a look here quickly. Okay. Just want to make sure that I can see all your comments. Okay, great. So I've just opened up all of the comments alongside as well, so I can have a look at that. Okay. All right. So what we're going to do is I'm going to jump right in, and I'm going to start off with plant hormones. Now, I know that a lot of us really struggle with this section, and I think the reason why we struggle with this section is we don't know how to explain the differences between geotropism and phototropism. So I'm going to start off with talking about phototropism. So we're going to talk about the top half of the plant, focusing in how auxins affect the shoots or the leaves and the stems. So I think the main issue that comes down to our understanding is we don't actually know what auxins do, and we don't know how they affect the growth of a plant. So I just want to remind you that auxins are responsible for growth, which means that they are responsible for cells elongating, which means cells sort of start like this, and then over time they get bigger and longer. They don't just get more numerous. So that's the one thing. The next thing that auxins are responsible is for apical dominance. And if you don't know what apical dominance is, that's why trees have the least foliage on the top and the most foliage at the bottom. And then last but not least, we know that auxins are responsible for tropisms, which is what I'm going to cover right now for you. That is when plants are sensitive, or not sensitive, they react to gravity or light. Okay, so those are the 3 functions of auxins. Again, we need to just keep some of these in mind as we go through this section. So I want you to also know that the roots and shoots respond differently to auxins. So if we go back up here, I want you to know that the shoots, so the shoot at the top here, they are not as sensitive as the roots are. And so what that means is because roots are very sensitive, they actually require a lot less auxin to grow. And that means that essentially, when we talk about the concentration levels, shoots need more auxins and roots need less. Now, don't worry, everybody. This lesson is going to go up on my channel, so you will be able to watch it when we're done in case you missed anything and you want to fast forward and just see, you know, some of my notes that I've been making. So don't worry, that is going to be up there for you. So let's talk about phototropism first. So phototropism, as we know, is in reference to how light affects the growth of plants. And I'm going to just put a little scenario here. So we've got sunlight, right? And this is the classic way they ask phototropism, is with some kind of light source. Maybe they have a plant on its own, or maybe they have a plant in a box, something like that. But let me give you the basics. So we are at the tip or the end of the shoot, right? And the auxins are supposed to be fairly evenly distributed through the apical tip, right? And by the way, that is where we make auxins, right? In the apical tip. Now, what happens is when there is light coming from one direction only, which means it's unilateral, so it's coming from one direction, what happens is all the auxins start to collect. I'm just going to draw it a little bit bigger because I need to draw some other pieces in. They collect all on the dark side. Okay. So our auxins move to the dark side. And that is because auxins are sensitive to light. So they actually don't like light. It's because light will slowly destroy auxins, so they move away from it and they collect on the darker side. And now this is the interesting part that I think we don't know and that we get wrong every time. And you'll see now when we do the root what I'm talking about. Actually, let me draw this one again. Our auxins affect the cells in shoots differently to roots. You see, when the auxins collect on the dark side, all the cells on the dark side start to elongate. Right? And I'm just going to pause for 2 seconds because there's something wrong with my comment section. I'm just having a look here quickly. Right? So there we go. Sorry, back in action. So the cells on the dark side, they elongate, but the cells on the light side, they don't elongate. So they stay short like this, right? And so what happens is, because it elongates on the one side and not on the other, it leans. I can't do a nice little lean over. Let's see if I can draw it a bit better here. Leans over towards the light. Okay, so it bends towards the light. And that is because these cells here are elongating and getting longer, whereas these cells here are staying short. Now, I'm going to give you some tips about how you're supposed to write this answer in the exam. Number one, you always need to acknowledge, are the auxins stimulating or inhibiting growth? Okay, that's the first one. Acknowledge that. So in this instance, it is going to it's going to stimulate elongation. The second thing you always need to do is tell me what the cells are doing on both sides. So you're going to explain what's happening. And I think I'm going to run out of page. Oh, no, we go. On both sides. And what do I mean both sides? I mean what's happening on the dark side and what's happening on the light side. If you do these two things, you will always get full marks for your exam and you don't need to worry about losing anything out. And I think that's generally what we do not so well, is we end up losing marks, right? Okay, so now what about if we need to go over, now what happens if we need to do geotropism, right? So what happens inside of the shoot? Okay, so I'm going to quickly go over the shoot and what happens in geotropism. And then I will have a look at the chat and see what you guys are saying and what you would like me to cover. Just remember, I've got a whole bunch of things that I am still going to cover later on. So, stick around for the section that you want to cover. So now I'm going to go on to geotropism. Right? And geotropism is when plants respond to gravity. Now, the thing is, roots, they respond differently to tropisms than shoots do. In other words, there's a different reaction. So I'm going to draw you a seed, because that's the most common way they show this. And this would be a shoot growing out, which is already curved up. But now here is a root growing out. Okay. So I haven't curved the root yet. So this is the shoot. This is the root. Now, what's interesting is that the root has a very different or opposite response. And I think a lot of us get this wrong. And so that's why I wanted to do it today. Was that we don't actually know that the auxins play a different role in roots. What happens is, in a root, the auxins collect on the side of gravity. So they collect down here because they're actually attracted to gravity. So unlike auxins in the stem, which are repelled by light, they move away from light, auxins will move towards gravity. Right? Okay, so they move towards it. They sink down to the bottom. Now, when they sink down to the bottom, and I'm just going to draw this bigger for us so that we can see what's happening inside the root. The auxins collect at the bottom. But now you'll see this is the opposite of what happens in the shoot. Roots don't like auxins, because remember I mentioned to you in the earlier on, they're very sensitive to light. So they collect at the bottom because it's sensitive to light. What happens is all the cells that are being stimulated, or should I say being touched by the auxins, there is no elongation taking place. Whereas the side where there is very little auxins, those cells start to elongate. And this is because auxins inhibit cell elongation in roots. Whereas in shoots, it promotes cell elongation. So what is the final outcome? We end up having a root that will bend downwards, right? So it bends downwards. Okay, so if we were to draw it up on here, you would just have an extension of the root going down. Now, again, everyone, I want to remind you that if you are doing a geotropic explanation, you are going to have to say whether or not the auxin is inhibiting or stimulating. So here was me mentioning inhibiting. And you need to tell me what's happening on the side of the auxins and tell me what's happening on the opposite side too as well. Okay, so I'm just going to set this aside now for 2 seconds. Have a look at your comments. I know you probably have loads of requests in terms of video topics that you want me to do. Don't worry, I probably am going to be doing something now, but I'm just going to check my live comments alongside. I'm just having a look here. Okay, so I'm seeing Endocrine. Let's see. Shazi, you're saying, is it that simple? Yes, it is literally that simple. That's all you need to do. Amalia, I suggest that you stay focused on the video and not on the comment section, please. I see reflex and reflex action. We could definitely do that. Negative feedback, human reproduction. How do we do balance? Roxy, I see your request. Human responses to the environment, endocrine homeostasis. So I feel like we're all on the same page, because I am seeing menstrual cycle, ear, eye. Okay, don't panic, everyone. All right, I can see all your requests. So conveniently, look at that. I've already prepared for it. We're going to do the ear, and I'm going to focus in on some balance for you right now. Okay, because I knew you guys would want to do this one, and I know it's one that we suck at. It's not so hot, right? So I'll do some of the ear now, and then we'll move on to some of the other topics that I know you want me to go over with. Okay. All right, so let's talk about the ear. The ear gives us a lot of hassles because we don't know how to explain certain things. I'm going to start off with focusing on the semicircular canals, which are these guys up here, and they are responsible for balance. Right, so you have 3 semicircular canals, and the reason why you have 3 and they all sit at right angles to one another is because you need one that tells you you're going up and down. You need one that's going to tell you you're going left and right. And the third one tells you if you're going forwards or you're going backwards. So that's why you have 3. And when we explain ourselves in exams as to why we have 3, we use these 3 to not only explain balance, but a change in speed and direction. So that's often what the question actually says. It says something like, explain how she maintains balance while she changes speed and direction. So this is where we are sensing speed and direction and where we correct balance. So how does it actually all work? So if I go back up to my little diagram a little bit higher, okay, we've got our 3 semicircular canals up here. And if we zoom in just a bit more, so I can show you closer up, there we go. This is the cross section of one of our semicircular canals. Now, you actually don't need to know very much detail of the semicircular canal, so please don't believe the hype. If your teacher has told you you need to know a huge detail about everything on the inside of here, no, you don't. If you are writing a provincial paper, you definitely don't. If you are writing a paper that your school has set, you might, because it depends on your teacher and what they want to test. But in a final, they can't actually ask you all the details of this on the inside. All you actually need to know of this diagram is the name of our receptor cell, which is going to tell us which direction we're moving our head, and the fact that endolymph is moving around stimulating the cristae. And that's it. That's all you need to know. So what happens is when you're moving your head forwards, backwards, up, down, left, and right, the endolymph sloshes inside here. So if my arrows are showing the direction in which the fluid is moving, it moves around the semicircular canal and it comes around here and it stimulates our cristae. Okay, so the cristae is our sensory receptor. It is the thing that is receiving the information, and that is the thing that is going to help us correct our balance. Okay, right. So how do we explain balance? Let me write out the key points for how in an exam do you explain balance. So step 1, you always need to acknowledge that there is a change in speed and direction. If they give you a very specific example in the exam, please insert the example they've given. Next, you're going to say this change stimulates the cristae. The cristae, everybody, are the sensory structures I just pointed out to you now earlier. Where are those cristae? In the semicircular canals. Next point, this stimulus is converted into an impulse. Converted to impulse. Now, at this point, you're thinking, sure, she's writing a lot there because, I mean, that's already 4 points. Yes, everyone. Normally, these questions are anywhere between 6 to 8 marks. There's no longer an essay question. So this is the kind of longer things that they want you to write. So stimulus is converted into an impulse. We're almost done. The impulse is sent to the the cerebellum. Okay. How does it get there? Via the auditory nerve. And last but not least, the cerebellum sends an impulse to muscles to correct balance. And that's it. That's how you explain how you maintain your balance. I know it is a lot of points, but yes, that's all we need to cover. Now, I know you also want me to potentially also do posture, which is the maculae found in the sacculus and utriculus. So I'm just going to leave this up for a few more seconds in case anyone is writing it down. But let's not forget that this will be posted again. As Pop asks, ma'am, what does the vestibular nerve do? The vestibular nerve is a branch of the auditory nerve. So in other words, if you want to think of it like this, it goes from the semicircular canal. Okay. So from there, we go to the vestibular nerve. And from the vestibular nerve, we still go into the auditory nerve. In other words, the auditory nerve is the biggest nerve that leaves the ear. We don't like to use the vestibular nerve in our explanation for finals. Maybe your teacher would like you to include it. And let's just say if you include it, it's not a bad idea, especially if your exam is set by your teacher. They can definitely want to award a mark for that. But at the end of the year, you actually just need to know it's the auditory nerve that fixes your balance for you. I'm just having a quick squiz. Somebody asked, must you know the nerves? You don't need to know the structure of nerves, by the way. And what I mean by that is how to make a very big nerve, like all the different layers. You just need to know the structure of a nerve in terms of a neuron. So you need to know the structure of a neuron, but you don't need to know the structure of a nerve. So that means you need to know what a motor neuron is and what's a dendrite and an axon and etc. etc. Someone's asking, we don't do the essay question. No, we don't. All right. And yes, everybody, when someone says here you can fix the video quality, guys, that's your video quality, not mine. So that means you need to change your settings. And yes, my audio is fine on this side. You guys need to adjust your quality settings on your video on your side. My video is being shot in 1080p, so there should be no problems at all. Okay, that's something on your side you can fix. Okay, let's not waste any more time. That was balance. I'm going to just erase this. And now we're going to talk about the utriculus and sacculus. So the utriculus and sacculus sit in this swollen area over here, and they are responsible for posture. Okay, and if I just scroll over here to the side, you can see what I'm talking about now. You have two, you have the utricle and the saccule, and inside of them you have their receptors called the macula or the maculae. It's just a plural versus singular. And again, you don't need to know anything else. All you need to know is the fact that there is endolymph moving around inside this part of your ear and the maculae. And that's it. Okay, everything else inside here is optional knowledge. I say optional because maybe your teacher wants you to know it for your internal exam, but for the final, you don't need to know anything else. Okay, so how does the utriculus and sacculus maintain your posture? Now, some of you don't know what posture is. Posture is whether you are upright or are you lying down? That's what posture means. Okay, so when I say posture, that's what it refers to. Okay, so how do we explain posture? Okay, so, oh, whoopsie daisy. There we go. So all we need to do, yet again, is we need to acknowledge that something has changed, like we did with the change in speed and direction. We need to say that the position of the head in space, which basically means space as in which plane are you lying down? Is it vertical or is it horizontal? That's what space means. So the position of the head in space has changed. Now we need to talk about who's responsible for registering that. This is going to stimulate the maculae or macula. Where do we find the maculae? In the utriculus and sacculus. This, and again, you'll see this answer is very similar. This stimulus is converted to an impulse, right? And that impulse is sent to the cerebellum. They both go to the cerebellum because remember that's where balance and posture is located. Again, via the auditory nerve and the cerebellum sends an impulse to muscles to restore posture. So this is what would happen if you were like lying down and now you want to stand up. That's how you are fixing posture and you're standing up again. So that is how you would explain it in an exam if it was like a longer like paragraph question. I'm going to go and have a little look at our comment section in the chat so I can see if there are any more queries on the ear specifically. And then I'm going to move on to our next section because I know you guys want to go on to other stuff. Somebody asks, is it a common paper tomorrow? So yes, if you are writing tomorrow, you probably are writing a common paper at your school. You can confirm that with your teachers though, but tomorrow is the official Western Cape common paper. The cochlea, people want me to go do cochlea. Don't worry, Lolita, this video is saved. It actually gets automatically uploaded when we're done today. Let's see. Anything else? Anything else? I'm just looking for any other ear questions before I go on. Let's see. Oh, gibberellins and abscisic acid. I just saw someone ask that about plant hormones. Yes, you do need to know what they do. I wouldn't spend a huge amount of time learning them. All you need to know is that gibberellins promote germination and abscisic acid promotes dormancy or seed dormancy. And the fact that one of them keeps a seed dormant and the other one causes a seed to grow. That's really all you need to know. And the fact that you can use auxins to stimulate growth in plants, like as an artificial growth hormone. People want to know hearing and hearing in ear defects. Okay, so what's interesting, everybody, is there's really only like 1 or 2 hearing defects that we need to know. And I know you're probably thinking of grommets, right? Everyone's so scared of grommets because I remember last year they came up in the final paper. and people were not prepared. Okay, so what is a grommet? So this is your tympanic membrane or your eardrum. And what happens is, if these are your ossicle bones, I'm just going to draw 3 shapes here to represent the bones. And then here is your cochlea there, right? Okay. This is the middle ear. There is a tube that runs down here called the Eustachian tube or Eustachian tube. Okay. So how is this linked? So essentially, if fluid builds up in the middle ear over here, it is supposed to drain down the Eustachian tube, right? It's supposed to leave because the Eustachian tube is there to maintain pressure in your ear. Okay, that's like when you like blow in your ears and you hold your nose and your ears pop. That is when you equalize the pressure in your ears, right? So what happens is some people, their Eustachian tube can get blocked. Either because of their age, when you're very young, your Eustachian tube can get blocked really easily. So there's a blockage here. Or you get an infection. And so now this fluid can't drain anywhere. So it builds up over here. And as this fluid builds up, it causes the ossicles not to move. So they can't move anymore because this is all filled with fluid. The fluid can't drain away. So what we do is we cut a little hole in the tympanic membrane, which is a grommet, and we hold the hole open with a little piece of plastic or a little piece of metal. And what that allows is for the fluid to drain out of the ear into the ear canal rather, so that there's no buildup of pressure. Now, grommets are useful because if you drain out the fluid, it means that the ossicles can move and you can hear. If ossicles don't move, like I wrote here, then you experience deafness. Because if the ossicles are not moving, they cannot transfer any vibrations from the tympanic membrane through the bones and into the cochlea. And so that's why we want to keep everything moving. That is the main ear defect that they talk about. They can also talk about deafness. Yet again, deafness is not in the exam guideline. So it's not in like prelingual and postlingual deafness. It's not in the exam guideline. But again, if you're writing the common paper, you're good. They're not going to ask it because it's not in the guideline, because they can't. But if you're writing a school-based exam, that's a little bit trickier. Maybe your teachers want you to know it, but I can confirm it will not be something that they can ask in the final exam, because remember, if it's not in the guideline, they can't ask it in the final exam. Okay. Right. I actually want to move on a little bit, because if I don't, I'm not going to get to everything. And I know some of you want me to do different homeostasis functions. So I'm going to do a quick one on the skin, because the skin is actually an overlooked topic. And I want you to remember that when we talk about the skin, we talk about thermoregulation. I'm just having a look at some of your comments, everybody. Sorry, I'm looking at the chat as we speak, so that's why the camera's moving a little bit. Somebody asked, ma'am, is your study guide everything that can be asked in a test? Yes. Yes, I do have the flu, everybody. You can hear my nose is blocked. All right, okay, there's no more comments. I know the endocrine system is something that you want me to do. Okay. All right. I don't know why people want me to go over the diseases and disorders of the ear. That's not in the guideline. So it shouldn't be something you're being examined on. But again, I can't tell you what your teachers are doing, because in the end of the year, that's not in the final paper. Okay. Thermoregulation. So thermoregulation is in reference to two things. You may remember vasodilation and vasoconstriction. This is the two ways in which your body maintains its temperature. You do dilation in hot and you do constriction in cold. And what are we constricting? So if we look at the diagram here, we are constricting these guys over here, the dermal arterioles. So what happens is, if it is, for example, a cold day, these arteries are going to constrict and get smaller, which means the blood stays in this lower level here near the fat in your skin. And these vessels down here are called shunt vessels. So on a cold day, the dermal arterial circular muscles will contract. It will allow less blood to the surface of the skin. More blood is kept in the shunt vessels near the fat layer, therefore preserving the heat inwards or closer to the core of the body on a cold day. Another thing that happens on a cold day is, in reference to this structure over here, this is a sweat gland. And on a cold day, you will sweat less because the blood rushing past here will be much less. So whatever water accumulates inside of the sweat gland will not heat up and evaporate. So on a cold day, it will not do that. But on a hot day, the water that does collect in your sweat gland, because there's lots more hot blood rushing past it, will then cause the water to evaporate out of the sweat gland. In terms of our arteries, on a hot day, the dermal arterioles will relax and dilate, hence dilation. More blood will rush to the surface. And because more blood rushes to the surface, you lose more body heat via conduction, convection, and radiation. In other words, it all just comes out of the surface of your skin and into your clothing, and you lose more heat on a hot day, which is, of course, what you want to do. All right. I'm having a look at the comments, everybody. Okay. I'm going to do an endocrine, and then what did I save next? Oh, look, it's the female reproductive system and the menstrual cycle. So we are going to do that next, so don't panic. Let's do another endocrine system because I know that was requested. And again, I know it's something that we're not good at. And endocrine and homeostasis overlap, right? So I can do both of them at the same time. Let's do carbon dioxide. Let's do that one. No, let's not do that one. Let's do aldosterone. Okay. So this is the endocrine system. And let's do aldosterone very quickly. Okay. So aldosterone is linked to the adrenal gland. So that is the gland that we are linking here. And aldosterone's purpose is to regulate salt. So that's how the two topics link. Here is the endocrine portion. Here is the homeostasis portion. We use the adrenal gland to maintain the salt levels. Okay, so how do we explain how this works? So let's say hypothetically, there is a decrease in salt in the body. Another fancy word that we see often is called osmolarity. Osmolarity is technically a word you should know, but maybe you've never seen it before. Do not panic. Osmolarity just means how many solutes or how much salt is in the blood. And in this case, it's a decrease in salt. So it's a decrease in osmolarity. So there's not a lot of salt in the blood. That is the stimulus. So from there, we need a receptor. Now, the receptors who pick up on this low level of salt are the afferent arterioles, okay? And these are the little blood vessels that go into your kidney. You may remember them from grade 11. That's why we can't forget our grade 11 work. Now, these afferent arterioles they are going to respond by secreting something called renin. Now, renin is a hormone. It is not, as you noticed, aldosterone just yet. We can't make aldosterone immediately. So what we do is we make like an in-between hormone. It's a secondary hormone that we have to make so that we can eventually get to making aldosterone. So the afferent arterioles are told there is not enough salt. Their response is they are going to secrete renin. Renin is a stimulating hormone. It's going to get this process going. Renin is going to then stimulate the secretion of angiotensin, which is another hormone that is still not aldosterone. So yet again, we are stimulating one hormone with another hormone. So we've got two different hormones that we need to remember thus far. Angiotensin is sent in the bloodstream. So this is now in the blood. Remember, this is all happening in the kidney. This angiotensin is then sent in the blood, and it is sent to the adrenal cortex, which, if you remember, if this is the adrenal gland, if I draw it for you, there are two layers, an inside layer and an outside layer. Okay. The cortex is on the outside and the medulla is on the inside. So we're going to the outside part. We send a message, the angiotensin, to the cortex. From there, the adrenal cortex is going to secrete more aldosterone into the blood, which means, yay, finally we get to aldosterone. So that's finally at the hormone. And the response to aldosterone being secreted is that there is an increase in salt absorption from kidney to blood. Okay, and that's the full little sort of circle there. I can zoom out a bit if you want to see it all in its entirety. Okay, so that is aldosterone and that is for salt. You would just write the opposite, everyone, if you had too much salt. So if there is an increase in salt, then the afferent arterioles will secrete less renin, which means they will secrete less angiotensin, and that will then inhibit the adrenal cortex to secrete less aldosterone, so that there is a decrease in the salt absorption from the kidney to the blood. So in other words, you just do the opposite of what I've written here if you have too much salt, or if you've consumed too much salt. I know there was someone who also wanted me to do thyroxine, so it won't take me very long, I promise. I'm going to just give you a few seconds to even screenshot this if you want, but you don't have to, because remember, this video will go up for you to view later. I'm going to quickly do thyroxine, and then we will do all the female stuff, because I know you got 1,000 questions on that. Okay, so thyroxine. So let's say your thyroxine levels drop, right? So they're low. Somebody needs to be informed of this. So this is the stimulus. Who is going to be the receptor? The receptor is the pituitary gland. The pituitary gland registers this drop. It notices it, and now it must make a corrective measure. It must fix the problem. So the first thing it does is it secretes thyroid stimulating hormone. So this is the first hormone we are going to secrete. In this system, there are only 2 hormones. Unlike aldosterone, there are 3 different hormones you've got to keep track of. So here's the stimulus. It comes into the receptor. The receptor is also the control center, and it makes a decision. I need to secrete more thyroid hormone, so I need to secrete thyroid stimulating hormone. That thyroid stimulating hormone goes off, and it is sent to the thyroid. The thyroid gland is the one that's on your throat, and it is now going to be stimulated via thyroid stimulating hormone. And it is going to now secrete more thyroxine. So there is our second hormone, because there are two hormones in this system. And the result of secreting more thyroxine is we actually start to make less thyroid stimulating hormone. And it kind of makes sense, because this was the message to say, please make more. Now that we have made more, the thyroid stimulating hormone will go down. Okay. And last but not least, when the thyroid levels go up now, cellular respiration increases. Why? Because thyroxine is linked to your metabolism, and it is responsible for maintaining your metabolism. Okay. All right. I promised that we were going to have enough time to get to the female menstrual cycle and what's going on there. Okay. So what you have here is an ovary cut in half. It's a cross section. And I'm going to run through what we're looking at now very quickly so you can follow faster. Over here, we have the original follicles. At number 2, we have the Graafian follicle. This is number 3, is ovulation. And here, between these two structures, number 4, I'm just going to put them in the middle here, is the corpus luteum. Okay. All right. So let me just move this up a bit. All right. So hi, Kieran. All right. So let's talk about what's happening here. So what we're doing is we are trying to take a follicle and we are trying to develop it to become a Graafian follicle. From the Graafian follicle, we want ovulation to take place, and then we want a corpus luteum to form at the end. That's the full cycle. Okay, that's the goal. Now, how do we do that? It's all of your worst nightmares. It is the hormone regulation of this system, so the menstrual cycle hormones. So let me explain. We change over to the different colors. So how do we actually grow a follicle? So first things first, follicle stimulating hormone comes from the pituitary gland to the ovary, and it stimulates the follicle to grow till we get to this point over here, to step 2, the Graafian follicle, right? That's where we're trying to get it. Now, as the Graafian follicle grows, I want you to know that it is producing a hormone. So a hormone is coming out of the follicle, which is called estrogen. Now I'm going to keep the colors so you can keep track of what I'm saying. Going back to FSH, FSH stimulates follicle growth. The estrogen that is being secreted now out of the follicle as it grows, it regrows and maintains the endometrium. So I want you to know these two things are happening simultaneously. The follicle is getting bigger, and as it gets bigger, it secretes more estrogen. The estrogen is sent to the endometrium, and the endometrium is now getting thicker and it's regrowing. Okay? Right. Now, step 2. Now that we have a Graafian follicle that has gotten large enough, we need to ovulate it. So the hormone that needs to be introduced so that this ovulation can take place is LH. So LH stimulates ovulation. Okay? And it does something else. Once ovulation occurs, so the egg is released, LH also causes the corpus luteum to mature. So LH, let's say, stimulates corpus luteum to mature. Okay, a lot of you probably don't know that that's its second function. Now, the corpus luteum is formed here at stage 4, and it's going to start secreting a hormone called progesterone. Progesterone is, of course, the pregnancy hormone. So make a note here. Progesterone makes the endometrium more vascular and glandular. Okay. So what I've done here is I have put the hormones in 2 different colors. In red, you have all the pituitary hormones, and in blue, you have all of the ovarian hormones. Okay, so that's where the hormones come from and what they do. Now, the final piece to this puzzle is me explaining to you the negative feedback between this, because I know that's ultimately what the problem is. So I want you to use some logic here with me. If follicle stimulating hormone is responsible for making follicles, do you think you can grow a follicle and also be pregnant with progesterone? The answer is no, you cannot. Okay? You cannot have follicles growing while you're trying to grow a fetus. Okay? So that means if FSH is high, then progesterone will be low. That is their negative feedback link. Okay? It's the main negative feedback link. And of course, vice versa. If FSH is low, it is low because progesterone levels are high. That is because you're either in the later stages of the menstrual cycle preparing for possible pregnancy, or you are pregnant and you don't want to be growing another follicle, because you cannot have 2 embryos growing at different stages. One can't be 6 weeks and the other one 6 months. Okay? All right. So they have negative feedback. And remember, negative feedback is like a seesaw. And the seesaw represents how when one hormone is high, the other one is low. And they seesaw backwards and forwards. One goes up, one goes down, depending on the needs of that system. Now, how do we get to this point of high and low FSH and low progesterone? Well, I also can't ignore estrogen. Estrogen is a really important role as well, because yet again, if estrogen is high, everyone, think about it, am I going to grow another follicle? No, I'm already trying to grow one. And as this one here is growing and making even more estrogen, the fact that it's growing and making more is naturally going to suppress FSH. In other words, cause FSH to go down. It's signaling to the pituitary gland, I'm already growing a follicle. We don't need another one. please stop stimulating more growth. So again, if estrogen is high, FSH will be low. Now let's talk about luteinizing hormone. Luteinizing hormone is only able to go up when progesterone is low. Now, why does this make sense? Okay, why do they have negative feedback with each other? Luteinizing hormone is the hormone that is responsible for ovulation. If you are ovulating, you are not pregnant. Therefore, progesterone is low. Step 1. Step 2. Progesterone is low because if you are ovulating, you haven't made a corpus luteum yet. There is no corpus luteum to make progesterone. So that means the levels of progesterone are low until after ovulation. So when luteinizing hormone is high, progesterone is low. The moment you ovulate, then all of a sudden, what happens? Well, the corpus luteum forms and progesterone levels increase. And if progesterone levels increase, luteinizing hormones will decrease. So you don't ovulate again. And remember, if progesterone is increasing, FSH must be also decreasing. Because remember, if we are pregnant or possibly going to be pregnant, do we want to grow another follicle? No, we don't. Okay, I'm going to have a quick squiz into the comments alongside. I know I see some of you saying you're going to go into load shedding. I'm so sorry for that. I had to also avoid my own load shedding for this video as well. It only came on just before. Let's see. I have a question here. Random question back to auxins. Does auxins cause cell division to increase? Yes, it can. I'm having a look at all your questions. Tato, I'm so glad it makes sense. Yay. I'm so happy to hear that. It's always the best response I can get. Somebody asked, ma'am, is progesterone only secreted by the corpus luteum? This is a great question. No. The corpus luteum is the first structure to secrete progesterone. After the placenta forms, so you've fallen pregnant, the placenta grows, the placenta starts to secrete progesterone and takes over from the corpus luteum. But remember, that's only if you fall pregnant. Okay, so I'll say it again. The corpus luteum is the first one to secrete progesterone, but then the placenta will take over and it will start to secrete progesterone so that the corpus luteum doesn't have to do it anymore. All right. Yes, everyone, the video will be posted later on. You can watch this again later and skip through to the pieces that you want to see. Okay, you want to do a graph. I'll do a graph quick, because I know that's another one that we all struggle with, and that's a question. Not a question. That is a request in the chat. Okay, here we go. How do you tell the differences on the graph? Right, so here is our graph. Right, ready? So first things first, I'm just going to quickly put in day 14 because that's the most important day when we talk about these graphs. So the very, very first hormone that's going to go up on the graph is always going to be FSH, and it's actually going to peak twice. So this is FSH. So FSH is always going to be the first hormone to peak. So it peaks once in the beginning and it peaks around day 14. Then let's talk about estrogen. Estrogen also peaks twice, but you'll notice it peaks after FSH and after ovulation. One peak, two peak. This is estrogen. Again, if I talk about how their relationship works, if we are growing a follicle, then the follicle goes up in hormone. But as the follicle gets bigger, it starts to make estrogen. So then estrogen starts to increase. Now, as estrogen increases, the estrogen says, we don't need another follicle, thank you. And so follicle stimulating hormone goes down. These are often the two most confused hormones on a graph if they're not labeled, because they've got two peaks. They both have two peaks. The key is FSH peaks first, estrogen always peaks second. If you don't have both of them on the graph, and so that means you can't compare them, please know that FSH peaks before and on ovulation or near ovulation. Estrogen peaks before and after. So actually, this is the key point. This one will tell you, is this estrogen? And this point will tell you, is this FSH? Because these 2 points are often confused with each other. Okay, moving on. Let's look at LH. LH is always the easiest. No, we want red. LH is always the easiest hormone because it only peaks once. And that's on day 14. That makes sense because that's the day you ovulate, and that is the hormone responsible for ovulation. And last but not least, progesterone is always a very low hormone, and then after ovulation, it comes up, and then it drops back down again. That's progesterone. Now again, this peak, it's only a single peak, just like LH only has a single peak. This peak only happens after ovulation. It makes sense. Why? Well, think about it. If we have used luteinizing hormone for ovulation to occur, it means that we are left with a corpus luteum. The corpus luteum is secreting progesterone, and progesterone is increasing, right? If no pregnancy takes place, then the line goes down. If pregnancy does take place, then I want you to imagine a dotted line here. Let me just add it in. Then what will happen is you'll get a line that will go over here. It might be solid, like solid line or dotted line, but this line I'm showing you here, this is the level of progesterone if you are pregnant. So if they ever ask you what clue in the graph tells you that someone is pregnant, that is the clue. All right, let's have a look at any more questions. Mel asks, when will the FSH and LH levels decrease? I mentioned it a little bit earlier, but they decrease when estrogen and progesterone increase. So if you put them in two camps, like this. Let's make it super simple now. Like that. So you group them together because that's how they work. They work this way. If these two are going up, then these two are going down. And now vice versa. If FSH and LH are going down, it's because estrogen and progesterone are going up. So that's how their negative feedback works with one another. Nokwazi asks, ma'am, is it also important to know the process of implantation and gestation? Yes, so you do need to know the process of going from a zygote to a morula to a blastocyst and how it implants. In terms of gestation, again, according to the guideline, you don't need to know the phases of gestation. So like 3 months pregnant, 6 months pregnant, 9 months pregnant. But you do need to know the layers around a baby, a fetus, so the chorion, the amnion, and what they all do. You also do need to know the umbilical cord and what's in it, so the vein and the artery, and the fact that they are slightly different in function to the other veins and arteries of the body. What else? Any more questions? Oh, a reflex arc. I'm so sorry, we forgot about that one. Okay, that's going to be our last one for today, everybody. Let's do a reflex arc. Okay. So, quick sticks here. Let's say, for example, there is a sharp pin, and I put my finger over it and I touch the pin. That information is received by a receptor or a pain receptor in my finger. That information is then sent to a sensory neuron. That sensory neuron sends that information to my spinal cord, which, by the way, is made out of interneurons. The spinal cord then makes a decision, which is move away from the sharp, harmful object. That instruction is carried by the motor neuron. The motor neuron then sends a message to the effector, which in this case is going to be the muscles in my finger. And what do I do if here is the sharp object? I have now pulled my finger away from the sharp object. And that is a reflex arc. How simple is that? Super easy, right? I know some of you maybe want me to go over the spinal cord structure. So just a quick sort of recap on the spinal cord. Sorry, it's going to take me a hot second just to draw it. I wasn't prepared, otherwise I would have just brought a diagram with me. Okay, this is a cross section of the spinal cord. So let me apply what we just did now to this picture. So what's going to happen is our information is going to come up here through the sensory neuron, also known on this side. This is a sensory neuron running up here, and this is in the dorsal root. That's what this is, the side. So this thing is the dorsal root, and this pink line is my sensory neuron. It now goes into the spinal cord, and this is where the interneuron is. A decision is made and sent down via this is, remember, a motor neuron. Let me just write sensory on this side so you know that that's the sensory one. So sensory, motor. And this goes down this route over here, which is called the ventral route. Now, you only have to use these like more technical words, this one over here and this one over here, the ventral and dorsal, if they're asking about specific structures inside of the spinal cord. It depends, but if you're doing a reflex arc question, then this is all you really need to know and say, the stuff up here. That's it. Whereas this is if they were asking you to like label structures in the spinal cord. Where is the sensory neurons? In the dorsal root. Where is the motor neurons? In the ventral root. Where are the interneurons? In the spinal cord. That kind of thing. All right, if you've just joined, you may have missed what we started off in the beginning of the video. So just to recap, we covered plant hormones, we covered the ear, balance and equilibrium. I also spoke about grommets. We moved on to thermoregulation. We moved on to homeostasis of salt, homeostasis of thyroxine. We moved into the female reproductive system, into the menstrual cycle, and now we're ending off with the reflex arc and the spinal cord. I'm going to call it for there. We've been on for about an hour or so. This is just a taste of what our live lessons are like when you are a member of my channel. And don't forget, everybody, if you are a member of Rescue Me, that is the name of the membership, then you are entitled to a free copy of my study guide. You simply have to find the link on your members only community page. And then you just send me a request and I can then give you access based on your membership name that you've used to sign up, if the two correlate. And then I give you access to a free copy of the study guide. And of course, there's so many other things like the videos that I do just for members, member requested videos, past paper exam questions, those kinds of things. That's also a separate playlist. And so I hope that you all found this useful. Good luck for tomorrow if you are writing the final, if you're writing the provincial paper. And this video will be posted on my channel shortly. So wait for the upload. It'll probably take about 10 minutes or so. And I will see you all again soon. Bye.